So guys, now let us talk about the formed elements in the human blood. And as we know, there are three formed elements as such, that is RBC, WBC and platelets, isn't it? So let us begin with the RBCs, which are also called by the name of erythrocytes, red cells, red blood corpuscles. Now remember, the first important point about all formed elements is that they are formed in the red bone marrow. So all these formed elements are produced inside the red bone marrow. So whether it is the RBCs or WBCs or platelets, this is the common source of production of all these blood cells. Now let us talk about RBCs in great detail. Now remember, RBCs, as far as the formed elements are concerned, this is the most abundant formed element. Okay. So the value or the number of RBCs in the blood will be more than the number of WBCs in the blood, which will be more than, which is more than the number of WBCs or number of platelets. This is the most abundant formed element present inside the blood. Now then there is some important value that you need to remember. The count is 5 to 5.5 million. Right, this is 5 to 5.5 million per millimeter cube. So this is the count, RBC count, normal RBC count. This is the normal value of RBCs inside the human blood. Now, what is the shape of RBC? Shape of RBC is biconcave, disc shaped. Right? Why do you require a biconcave and disc shape of RBC? So this will be, on one view this can look like this, and on other view you will see that if you are seeing it from the sides, it is looking like this. So this is the side view and this is the normal view. Like you take it in this way and you are seeing this. The other one is like this. Right? This is the side view, this is the normal view. Now this is how the whole thing is arranged. Right? This is the normal shape of RBC, which is biconcave disc shape. And why do you require this? So that it can maneuver, it can directly go through all these kind of small capillaries which are present inside the human body. So even the small thinnest vessel, it can directly go through this with the help of this kind of a flexible structure. So the biconcave disc shape will provide it some flexibility with which it can go through all the small blood vessels of the body. It can go for all the twists and turns which are there inside the blood vessels. So that is the importance of having a biconcave disc shape for RBC. Now, does it have nucleus? There isn't any nucleus. So you will say no nucleus. Now, this is the adult RBC in adult form. Right guys, so that means if you prick somewhere and you will start oozing blood, if you see the RBCs in this particular blood, any part of peripheral body, if you cut somewhere, you will start bleeding, and that area if you just see, then mostly you will get something called by the name of adult RBC. So the most common cells that are adult RBCs inside the blood. So what will you see there? There will not be any nucleus over there. Right, there will not be any nucleus inside the RBC, that is the adult form. Now inside the bone marrow, this RBC is formed. The RBC is developing inside the bone marrow. During the process of development, in the initial stages of its development, there are stages where RBC has nucleus. So in the developmental stage, RBC has nucleus, and during the course of development, after a particular stage, this RBC will lose its nucleus and then forms the adult RBC, which is present inside the peripheral blood, or normally which is there inside the body. But inside the developing stages within the bone marrow, there are stages where RBC has nucleus. Right? Now what does RBC contain? Hemoglobin. Right? This is something that we are supposed to know. Fine. Hemoglobin. So the normal value of hemoglobin is 12 to 16 gram per deciliter. So this is the normal value of hemoglobin in a normal human being. Now the value of hemoglobin is more in males compared to females. The females have slightly lesser amount of hemoglobin compared to males, slightly lesser, but that doesn't mean they are anemic. No, that's not correct. They are having slightly less amount of hemoglobin compared to normal adult male. And you see, infants, that means a newborn child has maximum value. These infants are having the highest value of hemoglobin, that is around 18 to 23 gram per deciliter. This value is not important, but just to tell you, infants are having more amount of hemoglobin compared to a human male who has more value compared to female. This is a normal feature. This is very physiological. There is no disease that are there inside these kind of people. Like, this is the very basic idea that you are supposed to have. Now, hemoglobin has two parts. It has a heme part. This is nothing but iron, right? This is iron. And you have a globin part. And this globin is made up of amino acids. It is a protein, right? So this is the normal human hemoglobin. So the adult hemoglobin in human being, adult hemoglobin, for hemoglobin let me write it as HB, it is alpha 2 beta 2. That means in the globin chain, in the globin part of hemoglobin, in the globin part, which is the protein part, there are two chains of each of them. There are two alpha chains, two beta chains, two beta, two alpha. This is called as adult human hemoglobin. Now it has iron part also. Now what is the before birth? The child hemoglobin is alpha 2 gamma 2. So this is present before birth, and after that the value of alpha 2 gamma 2 decreases. So this is also called by the name of fetal hemoglobin. Right? Fetal hemoglobin. This is present in the fetus stage. That means before birth. And after that it progressively decreases, and then as you become an adult, mostly you will be having alpha 2 beta 2, which is the adult hemoglobin. Right? So these are the very important things that you are supposed to remember. Alpha 2 beta 2 is adult hemoglobin. Alpha 2 gamma 2 is fetal hemoglobin. So we have discussed all that we need to know with respect to hemoglobin. Now the function of hemoglobin is transporting oxygen. Oxygen is transported with the help of hemoglobin. So that's a very important feature that we are all aware of. I don't think you need to write this in this particular notes because we are aware of that, isn't it? Now let's go through this again. What is the life span of RBC? Lifespan of RBC is a very important value that you are supposed to remember, 120 days. Right? And what is known by the name of graveyard of RBC? This is the spleen. How do you see an RBC in the normal blood? See, when you talk about an RBC inside a normal human blood, it will be looking something like this. See, this is how the RBC looks like. It is red in color. It doesn't have any blue outline. I just thought of making that. Okay, so this is how the RBC looks like, and this red color is because of hemoglobin, and in the center there is no color. So this is a pale area which is present inside the center, right? So this is how the normal RBC will look like. Now if you talk about this kind of a view, the hemoglobin will be somewhere around here and somewhere around here also. So when there is a folding here, in the folding, this folding will push the hemoglobin upwards, isn't it? So there is a folding inside. This folding, or this biconvexity, this concavity, whatever is happening here, this concavity is going to push up the hemoglobin to this area and that area. That is why the hemoglobin is concentrated over here as well as here. In the center, you don't have hemoglobin. So this is called by the name of central pallor. That means in the center you have a pale area. There is no hemoglobin over there. It appears whitish. I mean, it is colorless. That's it. Fine. And graveyard of RBC, that means RBCs are destroyed in the spleen. So after 120 days of its life, it goes into the spleen, and in the spleen this RBC is destroyed. So this is the basic idea so far as RBC is concerned. So I hope this much of information will be sufficient for you to crack all the questions that are asked with respect to RBCs in the exam, right? So let's proceed to the next formed element, that is WBC, which is a bit more conceptual. It's bit more content is there that you are supposed to memorize. Let us now talk about the WBCs inside the human blood. Now there are many types of WBCs. We are supposed to know certain features of all of them. Now the basic idea is to understand the total number of WBCs all together, all these things together, the total number of WBCs inside the human blood, and that will be 6,000 to 8,000, right, per millimeter cube. So that means if you take 1 millimeter cube of blood, you will have 6,000 to 8,000 number of WBC. But I would like to call it as 3,000 to 11,000. That is the normal value that I follow. But yes, we are supposed to follow what is there in NCERT textbook, and this is the value given in NCERT. So there is no second thought. Remember this as it is, right? So the WBCs can be divided into two types here. We are aware of that actually. So the first type is called by the name of, the first type is granulocytes. Right, granulocytes, what does that mean? Granulocytes, they have granules, they have some kind of granules, and the other one is those are called by the name of agranulocytes. They doesn't have any granules. That is why you are putting an A in front of that. That means there are no granules in agranulocytes, but there are granules in granulocytes. Let us talk about these types one by one, right? So if you talk about granulocytes, the first type of WBC that are coming under this is the most common WBC, also called by the name of neutrophils. Right, so this is the most abundant WBC, so I will be writing it as most abundant WBC. Altogether, if you take blood, the most abundant maximum number of WBCs will be neutrophils. Now we need to know the differential count. What do I mean by that? How much is the percentage of neutrophil among all the WBC? So that is 60 to 65%. So these are given in brackets in NCERT. We are supposed to know this very much thoroughly, right? So this is a very important value that you are supposed to remember. 60 to 65% is the percentage of neutrophils present, percentage of neutrophils with respect to the total number of WBCs. Now let us talk about that in much more detail. What is the function here? Function is phagocytosis. Right, I can call it as the first defense line. Whenever a pathogen comes into human body, these are the first cells, these are the very first cells that go there and attacks it. Whenever a pathogen comes into human body, these are some of the very first cells that goes there and attacks it. So that is why we sometimes call it as the first defense line, and remember these points about neutrophils. Now let us talk about the neutrophil structure and how is it normally seen inside the blood. So if I just talk about the neutrophils, it will be somewhat like this. So it has an outer margin, a plasma membrane, it has nucleus. Now this nucleus is multi-lobed. There are many lobes inside the nucleus. 6 to 7 lobes will be there, and these lobes are connected like this. So this is how the neutrophil looks like. There are many lobes and these lobes are connected. So it is a multi-lobed nucleus. What is the type of nucleus is a very important question. Multi-lobed nucleus. Right, and yes, this is how the neutrophils looks like inside the blood. Now with respect to, we are saying that these neutrophils are granulocytes, isn't it? So there are granules in neutrophils, but what are these? What is the speciality? It's a neutrophil, so it won't stain at all. So these are very fine granules. These are fine granules that will not take up any stain. So many a times you will not see any granules in neutrophils. It is very difficult to appreciate granules in neutrophils. They are very fine granules and sometimes they don't take stain at all. They don't take up any stain, so it will not be visible under the microscope very easily. But what is most characteristic is this multi-lobed nucleus. So in the textbook picture you will see that the neutrophil is having multi-lobed nucleus like this, and the granules are not very much visible. And that's it. That's about neutrophils. Now let us talk about the next type of WBC, which again comes under granulocytes. Now this cell is called by the name of eosinophil. Right, eosinophil. Now the size of eosinophil is slightly larger than the size of neutrophil. So if I just try to draw this eosinophil like this, it is a bit larger cell compared to neutrophils. Now this eosinophil has a two-lobed nucleus. So this is one lobe and this is another lobe. Big lobes are there and these two lobes are connected in this particular format. Right. So this is the eosinophil, and that is why we typically call it as spectacle shaped nucleus. Right. So this is called by the name of spectacle nucleus. Now the size is slightly larger than that, and there are granules which are red in color. So there are red shaped, or orange colored granules. I don't think I have a color that is in that format. Let me try to find an orange color kind of a thing here. I think this will work out. Now the granules will be very coarse granules. So these are coarse granules. That means larger granules. And I said that in neutrophils there were very fine granules, isn't it? And these granules take up red stain. These are acidic. It takes up acidic stain. So it is very large. It takes up the red stain, and it is this is the way that you will see the eosinophil in the blood under the microscope, right? The size is larger than neutrophil. Let me write that also. This is larger than neutrophil. Right? Now, once you know this, you are supposed to know the percentage. I mean, out of the total number of WBC, what is the percentage of eosinophil? This is 2 to 3%. Right? Now, the function of eosinophil, what is the function? Function, obviously, the moment you hear eosinophil, the only point that comes to your mind is allergy. So, it is a mediator of allergy. It is important in allergy and also in infections. What infections actually? Parasitic infections. So, allergy, infections, typically this infection is of a parasite. Parasitic infection, this eosinophil has a role in targeting the organism and destroying the organism. Very much importantly, you are supposed to remember a point. It secretes as a function, let me write, it secretes what important substance? Histaminase. Right, this is a very important sentence that I have written here. It is a cell that secretes inside the granules. There are many enzymes. One of these enzymes is histaminase, and it will secrete histaminase. And this was the question that was asked in 2020 NEET exam, this year's NEET exam. Right, this was a match the following question. This is a very important question. It secretes histaminase. Right. So this is all with respect to eosinophil. Let us go to the next one, that is basophil. What is important for basophil is that this kind of a cell, this is important for secreting, its function is secreting histamine, heparin, anticoagulant. It will prevent the blood from clotting, isn't it? Histamine is mediator of allergy itself. Serotonin. Right, so these are very important things that are secreted by the basophil. So the point that I would like you to remember is that histamine is synthesized by basophil, but histaminase is synthesized by eosinophil. So this is a very important sentence that you are supposed to remember. This was a previously asked question. Histamine was there in 2020 NEET exam also, in the same question that we have seen. So these two questions were the same questions. They were match the following type of questions in which both of these things were discussed. Now, very important point that you are supposed to remember is that this is the least abundant cell inside the human blood. I mean, least abundant WBC. Least abundant WBC. So it is very less, isn't it? So that is just 0.5 to 1% of total WBCs. You will see basophils over there. It is very difficult to get a basophils, right? So this is a, you have a practical exam in class, first year of your MBBS in physiology. So you have a practical exam. You are supposed to take your own blood and make a smear out of that, see it under the microscope, and they will ask you to focus a cell. Now under the microscope, you are supposed to focus a cell of your own blood. So they will never ask a question. It is very unlikely that they will ask a question on basophils, because many a times you will not get basophil at all inside the blood that comes out of your hand, because at that particular time it's just a very small amount of basophils is there, unless you are having a disease. Unless you are having a disease, if you are very well and you are going to the exam hall as it is, like normally you will not get a basophil very easily. You will find it here and there somewhere or the other, but during the time of examination you may not get a basophil inside your blood at that particular time. So they may not ask you. It is very unlikely for a question in first year to focus on a basophil, or normally in the college in which I study they don't ask. So least abundant WBC, it is very difficult to find it under the microscope, but you will get it. If you keep on searching for it, you will get it. 1 or 2 in that particular slide, you will get it. That's it. So this is how the basophil. Now, how does it look like? The basophil is a bluish kind of a cell. It has a bilobed nucleus like this, but the nucleus is very subtle. You will not be able to see the nucleus. Instead of nucleus, you will see, these kind of dark blue kind of granules that will cover everything. It will cover the nucleus also. So you will find it very difficult to appreciate the nucleus. So these are big coarse granules which are blue in color, right? So these are blue granules that will be covering the entire cell. You will not be in a position to see the nucleus even. It will cover the nucleus also. It is very difficult to find out the nucleus over there. This is how the whole basophil looks like. It has a bluish tinge to it. It is looking very much blue. This is the word meaning of basophil. right? So that's it. This is how the basophil looks like. Now, neutrophil, eosinophil, basophils are the three important granulocytes that you're supposed to remember. These are the only three granulocyte WBC. Let us talk about the agranulocytes now. So now that we are familiar with granulocytes, let us talk about the agranulocytes, or the cells where there are no granules inside the cytoplasm. If you see this, neutrophils had granules that cannot be seen normally. Eosinophils had red granules. Basophils had blue granules. Both of them were coarse granules, large granules. Now these are agranulocytes. The first one is lymphocyte. Right? This is the first type of cell. Now lymphocyte, if you see the percentage here, this is 20 to 25% of all WBCs. Now when you see the blood of a person, the most commonly encountered cell will be neutrophils. Among WBC, the most commonly encountered will be neutrophils. Then comes lymphocyte. So this is the second most abundant cells. Function, it is providing immunity, so it is responsible for providing immune reactions inside the body. So here remember one important very basic concept that you are supposed to know. Whenever an organism comes into the body, a pathogen comes inside the body, it breaches the epithelium and it comes inside the body, the first cell that will encounter it is the neutrophils. That is why we call it as first line defense amongst the cells, isn't it? So amongst all these kind of WBCs which is providing you immunity or defense, the first cell to come there is neutrophils. But if the organism is persisting inside the body for some more time, the second type of cell that comes will be lymphocytes. So that is why we will be writing it as chronic infections, or I can write chronic inflammations, right? So this is an important point to remember. Whenever you are having a chronic infection or inflammation, chronic means it is persisting for a long time. Initially, it will be encountered by neutrophils. Along with that comes the lymphocyte after some time when it is becoming chronic. So after some time, if the foreign body or the pathogen is persisting inside the human body, the cells that are going to come is the lymphocytes. So this, if you just ask me how this looks like, the lymphocytes are very much interesting. You have a cell like this, it's a round cell, right? And in this particular round cell, you are having a very big nucleus. It's a very big nucleus, a darkly colored nucleus you will be having, and it is this nucleus. Let me try to make a highlighter in that particular color itself, right? This will do the perform the function. Okay, so this is how the whole nucleus will look like, right? And you are having a peripheral cytoplasm, and that cytoplasm is very small. So you have a nucleus which is very large. It covers almost full part of the cell, and 90% of the cell is this particular nucleus, and beyond that comes a very small rim of cytoplasm. This is how the structure looks like. So this is the rim of cytoplasm that I am at presently shading now. So it has a very big nucleus and a very thin rim of cytoplasm, and this nucleus accounts for 90% of the cell. Is made by, it is actually is actually containing the nucleus, occupied by the nucleus, and the rest 10% is the cytoplasm. This is how the whole lymphocyte looks like, and as I said, it is performing the function of immune reactions. I mean, yeah, it has its own function in immunity, and that comes in chronic inflammation, right? Now let us talk about the types of lymphocytes, which is very much essential. It can be of two types: B lymphocytes, right? And you have a T lymphocytes, right? So I mean, just by looking at this, just by looking at a picture of lymphocyte, I will not be able to say whether it is a B lymphocyte or a T lymphocyte, but it is based on the function that they provide. It is based on the site at which they are present. Then you can understand that we are classifying it as B lymphocyte and T lymphocytes. Remember this concept very clearly. Both B lymphocytes and T lymphocytes are produced inside the bone marrow. B lymphocytes and T lymphocytes are produced inside the bone marrow only. They are produced inside the bone marrow. B lymphocytes will remain in the bone marrow. T lymphocytes will go to the thymus. So this goes into the thymus and they mature inside the thymus. So maturation is what is going to make the difference. See, the point is, it is not based on where they are produced. It is not based on where they are produced that we are classifying it into B lymphocyte, T lymphocyte. Both are produced inside the bone marrow. As I was telling you, all the formed elements are produced inside the bone marrow. RBC, WBC, platelet, whichever it is, all are produced inside the bone marrow. But maturation of B lymphocytes is in the bone marrow itself. After producing bone marrow, after producing B cells, these B cells, B lymphocytes will remain inside the bone marrow, while T lymphocytes will go to the thymus. From the bone marrow, it goes into the thymus and it will mature inside the thymus, right? So that is why we call it by the name of T lymphocytes and B lymphocytes, right? B lymphocytes will produce antibodies. How is it producing? We will be discussing that in the topic of human health and disease, right? And T lymphocytes will actually help in. There are different types of T lymphocytes actually that will be learned in a higher level in class 12th. You have suppressor T cells, helper T cells, cytotoxic T cells. These are T helper cells, cytotoxic T cells, suppressor T cells, etc. are there. Now, all of them have different functions, but basically I can say that this T lymphocytes helps B cells to synthesize antibodies. So T lymphocytes normally, in a very crude sense, I can say T lymphocytes will help B cells for producing antibodies. B lymphocyte will produce antibodies. These antibodies will come and attack over the organism which is coming inside the human body. That's it. So these are the basic things that you need to focus on so far as the lymphocytes are concerned. Now coming to the last type of cell, and that is the last type of your agranulocyte, which is called by the name of monocyte, right? Monocyte. Now the monocyte will actually account for 6 to 8% of all the WBC in the blood, right? These percentages are very much important because that is a direct NCERT question that they can ask you. Even in the practice test you will get a question like this. A lot of times they will confuse you with these kind of percentages. Now when I was preparing, they used to ask a question which you need to match the percentages with the cells. Now these are the largest cells, isn't it? These are the largest cells. They are even larger than RBC. They are even larger than any other WBC. They are obviously larger than platelets. So these are the largest cells that you will encounter. Now the point is that if I just ask you the function, then you will say that what is the function of neutrophils? Neutrophil, the function is phagocytosis. They will come initially and they will phagocyte. They will eat up the organism. They will eat up the organism. This will also phagocytose, right? So, a basic idea can be like this. Neutrophils are very small cells, very small cells. These will take a very bits of, if there is a big organism, if this is a big organism, the neutrophil comes here and takes bits of these organisms away. They will eat small bits of organism. This macrophage is a very big cell that will engulf the whole organism as it is. So a lot of neutrophils will in turn eat up a small organism one by one. I mean, it is cut into pieces and that is eaten by a neutrophil. I mean, lots of neutrophils, but one monocyte is enough for a big organism. It will just engulf it and go. So this is the basic difference between the power or the ability of these two cells. Now, if I talk about the structure of monocyte, it is a very big cell. In fact, bigger than any other WBC, and it has a nucleus like this. This is called by the name of kidney-shaped nucleus, right? This is a kidney-shaped nucleus. So I hope I will be able to color it better this time. So this is the kidney-shaped nucleus, or I can call it as the bean-shaped nucleus. Now this is how the nucleus looks like, right? This is the kidney-shaped nucleus. So let me write it here. Kidney-shaped nucleus, right? And this nucleus is actually, around 50% of the cell. So half of the cell is made by this particular nucleus, is occupied by the nucleus, rest half is the cytoplasm. While in case of lymphocytes, you said that 90% is the nucleus and only 10%, that peripheral rim of cytoplasm you could see in that particular cell, right? So these are the very basic idea that you need to have so far as these kind of cells are concerned. Let's go to further discussion. Now, let me teach you a very basic idea of macrophages. See, you know something on monocyte, isn't it? We have just discussed something on monocytes. We know the shape of the cell, the function of the cell. Now, the point is, monocytes are the cells which are present inside the blood, right? So, there are monocytes here and there inside the blood. Let me mark it with blue color, so that will be more appropriate so far as monocytes are concerned. So, these are very large number of monocytes inside the blood vessels. But the point is, many a times the pathogen or the disease causing organism or the pathogen or antigen will not be present inside the blood vessel. It can be in some tissues, isn't it? It can be in your brain. The disease causing microorganism can be inside your liver. It can be in the lung, right? It can be in the skin. So if it remains in the blood vessel, it cannot be in a position to go and attack these kind of cells that are there inside the tissues. It is there inside the blood, but it will without going away from the blood, without coming out of the blood, it will not reach the tissues. So what actually happens is something like this. The monocytes are produced inside the bone marrow. All formed elements are produced inside the bone marrow. Now, once these are produced inside the bone marrow, let me write it as BM. So from the bone marrow, these monocytes are coming into the blood, isn't it? Now in the blood, these are called by the name of monocytes. They will circulate in the blood for around 1 to 2 days. So for around 1 to 2 days, they will be present inside the blood. Now after 1 to 2 days of circulation, it will go into a tissue. So it will come out of the blood and it goes into tissues. Right? After it goes into tissues, it will start to change its form. It will start to change its morphology. It will start to change its structure, and it forms a peculiar, dangerous looking, very these kind of structures it will form on its surface, and it becomes a very dangerous looking cell. And these cells, we have a nucleus here. These are called by the name of macrophages. So what has happened here is that monocytes were produced from the bone marrow. It circulates in the blood vessels inside the blood. They are called by the name of monocytes then. But after some time, it goes into the tissues, and there it forms a different kind of a cell. It will transform itself. It will go for a makeover, and they form something called by the name of macrophages. These have the same function as monocytes, but inside tissues. That's it. That's the difference. That's all. If the organism is inside the blood, it will engulf it. If this organism is in a tissue, say it is in the skin or lung or brain or liver, wherever it is, it will be engulfed by macrophages. So that is why monocytes are present inside the blood. Macrophages are the same cells with a different structure. The same cell has undergone a makeover, and it has become a dangerous looking cell, a scary cell, scary looking cell called by the name of macrophages. So this is present inside the tissue. This is a good understanding that you need to have at this level. Now, depending on which tissue it is present, these have different names. Now, let us talk about certain names that you can remember at this level. If this macrophage is present inside the lung, okay, you will call it by the name of alveolar macrophages. Right? If it is present inside the lung, you will call it by the name of alveolar macrophage. If it is present in the liver, you will call it by the name of Kupffer cells. So what are Kupffer cells? These are macrophages inside the liver. That's all. If it is present inside the brain, I mean not in the brain, but outer covering of the brain, basically, you will call it by the name of astrocytes. If it is present inside the skin, what will you call this? These are called by the name of Langerhans cells. So these are certain typical names that you can remember. Depending on the site where these macrophages are present, depending on the tissue where it is present, it has certain peculiar names, and these are the names that you can understand. Okay, you can remember this. It's a good thing to remember these. You will be coming across these names in different chapters. Depending on the extent of discussion that you will have at this level, you will come across these. At least you will come across Kupffer cells and astrocytes in different topics. So okay, that's all. So now that we are well versed with the structures of these kind of cells, let us try to understand that in a real time image. So this is called by the name of blood smear. This is just for fun. This is just for curiosity purpose. Nothing more than that. Okay, this is not. They will not give you a picture like this and ask you what it is. They can directly ask you questions from the textbook images, which are very similar to this. If you can understand this, then the textbook becomes easier. NCERT becomes more easier because we are supposed to understand that image which is there in the bottom page, bottom part of that page. So this is what you have here. This is a practical exam question in first year as well as in second year also of your MBBS. So especially in first year, you have to take your own blood for that. You have to prick here and you have to take one drop of blood. You have to make a smear out of that on a glass slide, stain it and focus the slide, the cell that they are asking you to show. So if you see this particular image, you can see that there are a lot of cells in the background, isn't it? These cells are your RBCs. So as I was telling you, RBC has a biconcave disc shape, and if you see from the sides, the concavity can be seen like this. And since the concavity is pushing the hemoglobin in both directions, that is why you see that the central pale area, there is no hemoglobin in the center. Hemoglobin is in the periphery. This is where the hemoglobin lies. This is where the hemoglobin lies, not in the center. So this is how the RBCs looks like, isn't it? This is how the RBCs typically look like. In the background, all these cells are RBCs. Now, the bigger cells that you see over here, bigger cells are the WBCs. Now let us try to understand the types of WBC here. This is a type of cell. It has 1 lobe, 2 lobe, 3 lobe, 4 lobe. There are multi-lobed nucleus. This is the neutrophil. Right? This is a neutrophil. Even this is a neutrophil. This is a neutrophil. See, 1, 2, 3, 4 lobes are there. 1, 2, 3, 4 lobes are there. Neutrophil. Again, this is a neutrophil. So these are many neutrophils you can see. So just by looking at this image, you can understand that neutrophils are the most common cells among WBCs. It has a lot of, there are lots and lots of neutrophils everywhere. You will be seeing that, okay, inside the blood smear, inside the blood. So you can understand how abundant the neutrophil is just by this particular diagram. Now if you see this cell, it is slightly larger than the neutrophil. See, this is very large compared to the neutrophil. Comparatively, it is more large. And it has a nucleus over here, it has a nucleus here. These are two lobes which are connected. This is called by the name of bilobed nucleus, spectacle shaped nucleus. This is your eosinophil. If you see the cytoplasm contains reddish kind of granules. May not be appreciable much here, but yes, compared to the other granules over here, this is again a eosinophil. It is a big eosinophil, one lobe, two lobe connected. So this is another eosinophil. Right, and these are reddish granules that you can see inside the cytoplasm. You will be able to appreciate it with experience. Now the next thing is, this is your monocyte. Right, it has this particular kidney shaped nucleus. If you can appreciate that, then that is monocyte. Okay, now what else can you see inside this? See, this is a lymphocyte. This is a lymphocyte. I hope you can appreciate a very large nucleus which is occupying 90% of the cell, and a very thin rim of cytoplasm you can see here. A very thin rim of cytoplasm can be appreciated over here. This is a lymphocyte. Even this is also a lymphocyte. This is also a lymphocyte. Right? This is a monocyte. Kidney shaped nucleus, bean shaped nucleus. You can say that, isn't it? Now coming to this picture, this is how a basophil looks like. How do you understand this? There are a lot of granules, coarse granules can be seen, blue colored granules entirely filling the cell, that even the nucleus is not visible. It is full of granules, blue colored granules. This is why you call it by the name of basophils. Now what is this? This is a neutrophil. Right? So 1, 2, 3, 4, 5 lobes are there, and these are connected. That's it. So this is how you will identify the cells over here. Now try to go for the textbook diagram, and with this I can assure you that you will understand this. Understand if they give an image based question, which type of cells. This is very much important to know this at this level. The characteristics of cells are much important, right? The last type of formed element here, and that is your platelet or thrombocyte. So this is something that promotes thrombus, or it helps in clot formation. Right? So clotting is there are 1 or 2 paragraphs to be learned in this textbook. We don't have to go into so much of details because it is a vast area. Now platelets, yes, if I start talking about that, where is it formed? Again inside the bone marrow. All the formed elements are present inside the bone marrow. It is formed inside the bone marrow. They are formed from cells called by the name of megakaryocytes. They are very big cells, megakaryocytes. Where are these located? These are located inside the bone marrow, and it is from these megakaryocytes it will start producing platelets. So megakaryocytes are very big cells. It has a very big nucleus also, and from this the small platelets will bud off. So there is budding of platelets like this. So it's a small piece of that will be broken, and that will come out as a platelet. And this is how the platelets are produced, from megakaryocytes, which are present inside the bone marrow. And platelets will release certain factors. These factors that are released by the platelets will help in clot formation. That much importance is there. I mean, this is only what is required at this level. Now what is the normal value? This is important. The count, platelet count is 1.5 lakh to 3.5 lakh per millimeter cube. And what will happen if the number of platelets are decreasing? What will happen after that? If there is no platelets, or if the number of platelets are decreasing, clotting will be prevented. So if this kind of a patient is undergoing some kind of if there is a trauma to this patient, if there is an injury, if he will profusely bleed, so if there is a deficiency of platelets, there will be uncontrolled bleeding, and yes, it is a serious kind of a issue, and that has to be managed accordingly. So this is how the entire discussion of our basic idea of the formed elements are coming to an end in this particular video. So we have said everything from keeping in mind all the questions that were asked in the recent years. Now I hope if you try to solve all questions after this, you will be able to solve at least 99.99%, and I have considered everything that is there in NCERT textbook, so we will not miss any question in the NCERT. The rest questions that you will not be even in a position to solve will be advanced questions that are not taught to you in this particular level, so we don't have to consider so much of that. Try solving the question papers that are asked as practice test, and if there is something that is not taught in this particular lecture, you can add this up in this particular notes. You can keep on writing those points inside the textbook, and I don't think these are much important here. Now with this much of information, I hope you will be able to solve all questions from the previous years. I have considered that because this is a very important area, a very high yielding area so far as this chapter is concerned. So we'll meet in the next lecture with the next topic, that is blood grouping. Thank you.